objective and activities. And the products, products metabolites, chemicals, and some products like DNA and others, others. And when we're talking about industrial microbiology and the benefits, the benefit effect of some of these microorganisms, find out that microorganisms that we use in industry can also be used to degrade organic wastes. They can be used to detoxify industrial wastes and toxic compounds. They can use to degrade petroleum and to manage oil spills. Some bacteria are being used, some organisms are being used to do that industrially. So industrial microbiology also encompasses activities like production of plant control. Agents, inoculants, and all those things that use advanced fertilizer are part of industrial microbiology, a process whereby microorganisms are being used to produce a product for optimal gain. So as I told us earlier, the organisms that are going to be must be isolated from nature. They be screened for product production. They be genetically modified for product use, and there's a way to make for conscious maintenance, not like one we use in the lab. There's a way they normally maintain their cultures, and then they now produce a mass culture of that organism. So they are using their bioreactors, and of course they recover the products, and then there's a process called purification. So what are the properties of a useful industrial microorganism? That is, the organism that can be used industrially from industrial microorganism must have some qualities. One, the organism must be capable of growth and product production in a large-scale culture. The organism must be capable of growth and product production. That means it's something that should be able to amplify and at least give the industrial people are only interested in gains. Put little effort and massive production so that they can get their cuts. So the organism must be the one that is capable of growth and product production in a large scale. Secondly, this organism should produce spores. If it's a fungi or yeast, you should be able to produce some spores of some other reproductive cell so that it can be easily manipulated in light vessels to grow on an industrial scale. So it should be a organism that is actively, fastly multiplying, not the one that has slow growth. One that's small thing like this, it just multiply. And of course, the organism will be able to grow rapidly and produce the desired results in the industry in a very short time. Number 4, the organism will be able to grow in a liquid medium at a very cheap price. This is a liquid medium that is not expensive. The organism will be able to grow in that. Number 5, the organism that is going to be used for industrial microbiology as a rule must not be a pathogenic one, because pathogenic organisms only cause disease.
I said you should come and enter.
It must not be a pathogenic organism that is infective to man or animals, because of the high number of people that are involved in the production and what it involves. Pathogenic organisms are not used in industry. And of course, number 6, that organisms that are going to be used for industrial microbiology must be amenable to genetic manipulation. I've been saying it. There's genetic engineering that is done on that organism, to make it better than it is, and to give them maximum use. And of course, the organism must be genetically stable.
How do you use that?
Must be genetically stable. Now, how will this organism be isolated? The first step in producing an organism for industrial microbiology
Let me give you one. Come and
is
pick one.
to develop a producer strain
It's me that want to eat it. So, but since you've seen it,
that will lead to the isolation of that concerned microorganism from the national habitat. And natural habitat is usually soil. So, or maybe you get it from some of these companies that used to produce or maintain pure culture, like the American type culture collection, ATCC. Some of us that have done researches on some of these, some high class research, found out that the organs you're going to use as your control must be a regulated one. And many times in this company, in particular the one we use the ATCC organ. So the microorganisms of industrial importance are generally bacteria, actinomycetes, fungi, and algae. And they occur virtually everywhere. You see it in the water, you see it in the soil, you see it in the surfaces of plants and animals, plant tissues. But the most common sources of some of these industrial microorganisms are soils, lakes, and river mouths. So the first step is to be able to isolate the organism. And oftentimes, the ecological habitat from which a desired microorganism is more likely to be isolated will depend on the characteristics of the product that is derived from it. That is, the type of thing you want to get from that organism will determine the type of habitat you can get it. For instance, if it's an objective to isolate a sort of enzyme, we can resist high temperature. The obvious place to look for to get an organism that can be used for that is from hot water springs. Secondly, the organism must be screened. We call it screening of microorganisms for new products. This is a set of selective procedures that allow for detection and isolation of an organism that is produce or that's able to produce the desired metabolite. I mean, what you wanted to do, what you want to get, you screen for it in that organism. And ideally, as for industrial purposes, we talk about costs. One, they not spend so much money on. So the screening for this organism will be rapid, inexpensive, and predictive, but specific. You don't need to, they don't like just wasting money like we're doing in the laboratory on science, on research. You work on a lot of things to get something. No, they want something that get results. Work on it and to get them what they need. So it must be screening for them must be rapid, inexpensive, and predictive, and specific. So that's why industry people, because they are interested in what they can get, they didn't want to bother on. So that's why on working on to get something, they always work on on finished products. And the third thing we discovered is that there's something called inoculum development. That's the preparation of the population of organisms from a dormant stock culture to an active stage of growth that is suitable for inoculation to the final production stage. This preparation of that's the type of organism that you needed from a relatively unknown culture to a point whereby they can be easily inoculated to achieve the final production stage. So, and this process is done using flask cultures, and that flask culture can be a flask culture of about a few liters to tens of liters capacity because it's industrial. And of course, the first stage is the preservation of the organism. And the way they preserve the organism in industry is not like the way we do it in the conventional lab. Because once an organism has been selected or created to serve that purpose, it must be preserved in its original form for further use and study. Because when they are doing something we call the culture, it can lead to mutation. When they are recording also in lab, if you are, for instance, old cultures of this are more smaller than fresh one because a mutation has taken place. So in the industry, what they normally do is that they usually do something called periodic transfer of cultures because most of those cultures, by the time you are recording, the mutation will take place and there is something called phenotypic changes that lead to genetic disturbance, so to say, in the organism will take place. So what they do is that they preserve this organism in a natural state, in a good way, so that nothing about that organism will be lost. And the best way to do it, they can lyophilize it, they can explain, they can do it, they can take it through pre-drying, or they can store it in liquid nitrogen to preserve the organism so that next time when they want to use it, just take an ample of it, and of course, you go through the process. Before you know it, the product is on. Instead of the culture. And of course, how did this process optimize? The process are optimized by the use of something we call bioreactors. A bioreactor is a device in which a substrate of low value is utilized by living cells or enzymes to generate a product of higher value. And these bioreactors are extensively used for food processing, fermentation, waste treatment, etc. And on the basis of agent use, bioreactors are divided into two categories. There are bioreactors that are based on living cells, and those that are based on employing enzymes. But in terms of process requirement, they can be aerobic bioreactors, they can be anaerobic bioreactors, they can be solid state bioreactors, they can be immobilized cell bioreactors. And when I'm talking about the bioreactors, it's all we do with heterogeneous system. You have two or three phases, two or more phases, can be liquid, gas, or solid. Therefore, optimal condition for fermentation necessitates efficient transfer of mass, heat, and momentum from one phase to the other. So that's ends our lecture on applied microbiology. That's public health microbiology. We talk about the aspect of food and industrial microbiology. I don't know whether there's any question from the virtual class. Any question, please? Hello? Is there any question?
No question, sir. For now.
You said?
No question.
No question. Okay. So I will send the lecture notes to the class rep, and I have some review questions that I will also give alongside with the notes, which I will want you to send to my line, the form of continuous assessment. I think understand. There's some things that will go with it. So thank you very much for your patience. I'm sorry for the glitches we experienced in terms of communication. Of course, we are still growing, and God will help us. Wish all of us the best of luck. God bless.
Yes, sir. Thank you, sir.
Thank you, sir. And God bless you, sir.
Thank you, sir. And good night.
Hello? I'm going to end
Hello,
this
sir.
session. I see, I asked whether there's any question. Because there's some voices on that ground. I'm not sure I just said. Let me see. Yeah. After 22, I'm over. So Auntie, sorry for your taking your more than 19. Oh. Yeah, let's see. Okay.